morning students. Today we will discuss about development of pollen grain. Yesterday we have discussed that microsporangia has four walls, that is epidermis, endothecium, middle layer and tapetum. Epidermis, endothecium and middle layers are protective in nature, whereas tapetum provides nutrition to the developing pollen grains. Each cell has more than one nucleus and dense cytoplasm. Now, microsporogenesis. Microsporogenesis means development of pollen grains from sporogenous tissue. Each cell of sporogenous tissue, also known as microspore mother cell, divides by meiosis to become four haploid cells called pollen tetrad or microspores. Pollen tetrad are also known as microspores. Then there will be a further development of a pollen grain. Now each microspore divides unequally. Each microspore divides unequally and forms two cells. One larger one is vegetative cell and other one smaller one is generative cell. This generative cell becomes spindle shaped and floats in the cytoplasm of vegetative cell. This generative cell floats in the cytoplasm of vegetative cell. Then this generative cell divides by mitosis and forms two male gametes. It means the two male gametes are produced by this generative cell. Now pollen grain. Now there is a development of pollen grain. Pollen grain is covered by mainly two walls, that is inner wall and outer wall. Inner wall is known as intine and outer wall is exine. Now first one is intine, that is inner wall. Intine is made up of pectin and cellulose. This inner wall is made up of pectin and cellulose, whereas outer wall, that is exine, is made up of sporopollenin. This sporopollenin is a resistant organic matter. This exine is absent and at that location it is known as germ pore. The location where exine is absent are called germ pores, from where pollen tube grows when it falls on stigma. Beside this, some people are allergic to pollen grains and suffer from bronchial disorders, which leads to asthma or bronchitis. Pollens are also considered to be nutrient rich and are used as supplements to enhance the performance of horses and athletes. Pollens lose their viability from minutes to few hours or months, depending upon variety of plants. That is, like example, pollens of rice and wheat are viable only for 30 minutes, whereas those Rosaceae, Leguminaceae, Solanaceae family remains viable for months. The viability of pollens can be increased by cryopreservation.